Sorry, let me link it in the chat. So this is top-down control of hippocampal signal-to-noise by prefrontal long-range inhibition by Ruchi Malik, Yili, Selin, Schamiloglu, and Vikaas Sohal. Sohal?
Yes.
I'm sorry.
I don't know. I don't know any of
Where are
them
they from?
personally. UCFS. Am I saying this right?
Okay, cool. Please,
Yeah.
Yoshi, tell us about what top-down control of the hippocampus does.
Well, it directs novel object recognition in mice. So the idea is that the idea in humans would be, I found a very nice popular science article too, that says like if you park your car at a parking lot and you're trying to remember where your car is, you will just pay some attention to some objects that are close to your car so you can find your car later again. So you pay selective attention. And this paper now found a connection between the PFC that usually directs attention and like kind of is the
Garbage
governor,
bin off the brain.
the garbage bin of the brain, but also one of the tasks as garbage bin of the brain is to do the executive function, right? To say what is most important at the moment. So this is an example of that.
Is that what the PFC does?
PFC does planning and attention. I mean, the PFC does attention. So this is a specific pathway to the hippocampus that tells the hippocampus to pay attention to certain things
Right, and how does it
in
work?
the environment. It works very interestingly because I was if you read this episode, I was so confused. So it is a long-range inhibitory connection, usually inhibitory neurons
really?
in the cortex. Yeah, usually inhibitory neurons in the cortex are local interneurons that
Yes.
inhibit like pyramidals and other interneurons just around them.
May I interject? I'm a striatum guy.
Yeah, I said in cortex, okay?
Yes, subcortex. Okay, fine. It's true, I suppose.
In cortex they do this, but in striatum they are more long range, right? Or not?
The striatum output neurons are GABAergic and inhibitory, and
Yes.
they have long-range projections that are inhibitory.
They are long range.
I'm very used to long-range inhibitory projections.
Yeah, but in cortex, like it's not.
Yeah.
No, it's not. There are not so many
Wait,
examples.
but they're not the pyramidal cells then. They're like, what are they? PV neurons or?
Yeah, so I didn't find out what. Maybe if we dig deeper, they wrote it somewhere, but they don't
VIP
write so. Wait, they don't write so clearly. So there's a connection from PFC to hippocampus, and they project to VIPs in the hippocampus, as I understood. But I don't know what the type of interneuron is, because it's not an interneuron even. So maybe they didn't even bother. Maybe they just said like, yeah, we found some inhibitory neurons, and they are long range.
The graphical abstract is a bit small, but you can see they indicate that it goes to the VIP neurons. What's that? Vasoimmunopeptide something?
Yeah, they write. I cannot say it either. They wrote it in the abstract. Full length. Vasoactive intestinal polypeptide expressing interneuron.
Basically what I said, yes. It's just a marker to identify a specific population of neurons. They're pretty common in the cortex. I think you find them in the prefrontal cortex as well.
Yes, you find them all throughout cortex.
So theoretically, these could be VIP neurons in the cortex projecting to VIP neurons in the hippocampus.
Yeah, but I don't think they say what is like the marker in the projecting neurons, right? They don't write.
Yeah, because I'm trying to read through it, and they just say long-range GABAergic neurons, LRG neurons. Yeah. That's how they describe them everywhere.
But VIP neurons are used to identify a subpopulation of interneurons, so locally projecting GABAergic neurons. So it makes sense to identify those as VIP because this has been used like a lot in literature. But like for the long range projecting, maybe it doesn't even make sense to identify them because this is not a. Yeah, it would be interesting, but it's not like a requirement.
Yeah, I mean, I would be very curious. I would be very curious to know what population this is. Like, do they correspond to any of the known inhibitory markers? I mean, they must have VGAT, but Do they have
So
PV, somatostatin, VIP,
yeah, so
TH?
we have to double check the whole paper, and maybe they didn't find it. Maybe they don't. Maybe they have to do sequencing to see
That's
if
a
they
little...
express.
like,
Yeah,
could
but
this
if
be
you
a completely new, novel population of neurons in the PFC? Because
yeah, because
that's...
if they're long range projecting, they could be.
I mean, that explains why it's in cell.
Yeah, bro. So, but I didn't read everything in the paper, right? So I'm not sure if they describe this or not. Maybe they describe it somewhere in some little corner in that
Mm-hmm.
figure.
Yeah, no, I mean, I would
Um,
expect this information to be in the abstract. If it's not there, then
yeah, I
it could
don't
be a
think
badly
they.
written paper, but they probably didn't identify the
Yeah.
neurons. Interesting. So
Um,
how does it work? So you have inhibition on the... because you want the PFC does something, it becomes active, and then it inhibits the
yeah.
hippocampus, and that increases attention somehow?
No, it's so wait. It inhibits the VIPs, but VIP neurons are
Mm.
usually disinhibiting interneurons. So they disinhibit the rest, or like the VIP neurons inhibit the rest of the local interneurons in the hippocampus,
Yes.
and that means they disinhibit like the overall activity. They disinhibit the pyramidal neurons. So there's like an inhibitory long range projection, and that makes more activity to the in the place where it projects to. So this was like super confusing. I was
No,
reading
no,
the
no, no.
abstract like
I think you're missing one step because
inhibition
it's...
of
no, because
inhibition of
it's not
inhibition.
disinhibition. It's
No,
disinhibition.
yes, I know, but they write disinhibition in the abstract, so I was like super confused.
Right, right.
But it's this disinhibition, yes.
So it's excitation. No, no,
It's
it's inhibition again.
excitation.
It's inhibition again.
I don't know. There's more
Because
activity when
you
you
have
act.
the...
When
so
you
wait,
activate
wait.
the thing. Yeah.
Let's take it one step at a time. So you have a pyramidal neuron that fires. It's inhibited by an interneuron. So if the interneuron is active, there's less activity.
Yeah.
And then there's the VIP neurons, which inhibit these. So if the VIP neurons become active, there's more activity. But they're inhibiting the VIP neurons, so there's less activity again.
Huh? Wait.
I think there's three
Oh,
steps.
there's less activity.
There's less
Oh,
activity in the hippocampus.
did I still mess it up? Damn it! I thought about this and I'm still messing it up.
They don't really...
Oh, sorry. No, it reduces hippocampal. Yeah, no, you are right. Sorry, it reduces hippocampal activity when you
Yeah.
activate this pathway.
And the net effect of these actions is to specifically enhance the signal-to-noise ratio for hippocampal. This is interesting because I've read a paper on somatosensory cortex where a very similar thing happens. Basically, say you have a very noisy environment and somebody's talking, and the person who's talking is louder than everyone else, but you can't hear them because there's too much background noise, and you just quiet everything down, then the person that's been talking is also quieted down, but the rest is just... it becomes completely silent, so then, that one voice stands out. So it enhances signal to noise by inhibiting the entire network.
Oh, this is a good example. Yeah, because there's a lot of... in the cortex, there's a lot of inhibition going on to like enhance the strongest signal.
Especially in the superficial layers of cortex, the input layers. This is it's a relatively novel phenomena. I was talking about the lab of Adam Cohen was looking into this at some point. We're doing voltage imaging in the cortex.
I don't think it's like all these mechanisms are not like super clear. There is this idea of signal to noise ratio and inhibition improves it, but it's not like worked out how exactly it works. So these are very interesting studies then, right?
Yeah, and the other problem is, or the other complication is, we say, okay, it goes from this neuron to that neuron to that neuron to that neuron, but there is connectivity between these too.
Oh, yeah, like the local circuit does a lot here, and we don't, like, this paper does not describe everything that happens in the local circuit, so it's...
But yeah, very interesting work. I would really love to know the identity of these. Well, let's not go that far. I would really like to know the identity of these inhibitory neurons, but Yoshi really likes the PFC, so he's been traumatized. Shall we go on?
Yeah, let's go on.